In this video, we're going to talk about a process known as protein synthesis. To synthesize means to make, so saying protein synthesis is just a fancy way of saying how our cells make protein. This is tied to genetics. This really is what genetics is, so recall that your gene is just a recipe for a particular trait. And when we say it's a recipe for a trait, what we mean is it's a recipe for protein, and that recipe is carried in our DNA. Even before you took this class, you probably knew that DNA controlled your cells, so at least you hopefully knew that the nucleus was in charge, and now we know that DNA is in the nucleus. And so how does DNA control our cells? Well, they control our cells by controlling proteins, because proteins do most of the jobs that cells does. It carries out most of their functions. When we talk about protein synthesis, what we're really talking about is how DNA functions. So let's remind ourselves a little bit about DNA so that we can help understand how the structure of DNA is related to the function. So the first thing we need to remember is that DNA carries a code or a recipe, and the part of the recipe that matters is the order of the bases. So remember, DNA is made up of nucleotides, and the part of the nucleotide that makes up the backbone is the same, but the bases that stick out in the middle vary. There's thymine, adenine, cytosine, and guanine, or A, C, T, and G. And the order, so for example in this picture it's T, A, C, T, G, C, C on the bottom and onward. That order, the sequence of the bases, is what carries our code. While the backbone never comes apart, therefore maintaining the sequence of DNA, it is important that we be able to pull apart the two strands, because that's how we copy it for replication. That's also how protein synthesis works. So the way that the DNA, the two strands, are able to be pulled apart is through hydrogen bonding. If you recall, hydrogen bonds are very weak individual bonds, but in number they're very, very strong. If we can go and snip them apart one at a time, they should be relatively easy to separate. That's how it works in replication, when helicase goes and breaks apart the bonds between the two strands, and that's how it'll work in protein synthesis as well. The last thing about DNA we need to remind ourselves about is base pairing. Remember, the order of the DNA is what carries the code, but DNA is only able to copy itself because of what we call complementary base pairing. So hopefully you remember from earlier in this unit, adenine always pairs with thymine, and cytosine always pairs with guanine. And so if we know one side of the strand, or if our cells have one side of the strand, they can construct the other through complementary base pairing, or A's with T's and C's with G's. So in this model, I've assembled a strand of DNA. Notice on the DNA in my model, the rounded bases represent the sugar. The arrows represent the phosphate. Notice the phosphates are pointing in two different directions. The top strand is going to the left, bottom side's going this way. And notice I have complementary base pairs. So this is A. It should bond with T, and notice that snaps in there like this. It would not work on G, and it really wouldn't work in there on C. So that's all about DNA structure, but how does it actually work? How does DNA function to make proteins? So before we go on, I just want to use a little bit of analogy. Remember that each cell has a complete set of DNA, but only one set. And that one set controls everything that cell will do in its life. And so the cell has a nucleus for the explicit function of protecting the DNA. And so just like if you've got something really special you don't want it to be passed around, that's kind of like the DNA. So my grandma, what she was known for was shortbread. She was Scottish, and she made like this most buttery sweet shortbread on the planet, and it was amazing. And so in my family, her recipe is kind of like a family heirloom. And when all of my cousins and I got older, we wanted a copy of it for ourselves. Now my grandma's been gone for a long time, but we have a couple of copies of her handwritten notes about how to make this shortbread. Now when my mom and my aunt were passing it on to my cousins and I, they didn't want to give us their copy that grandma had written them of this DNA, but we still wanted the recipe, so they made a photocopy of it. Nowadays we take a picture of it, and they gave it to us in that way. And DNA is a little bit like that. DNA is this special, very important bit of instructions, and we want to keep it protected and safe, just like my mom and my aunt keep it protected in a recipe book in their kitchens. And to make it, we don't need the exact original, we just need to know what the instructions are. That's how protein synthesis works. So before we can move on, we need to understand the operators in protein synthesis. So DNA has the instructions, but all of the activities are carried out by a structure known as RNA, which is another type of nucleic acid. So DNA and RNA are cousins. They're both made up of nucleotides. They both have a helical structure. In other words, they both twist. But DNA is a double helix, RNA is only a single strand. DNA has the bases adenine, cytosine, guanine, and thymine. In RNA, adenine, cytosine, guanine are all present, but instead of having thymine, there's a base called uracil. Now, uracil and thymine are chemically very, very similar. There's a little bit of difference, and that is this methyl group. You don't need to know that, but just that wherever thymine would be, we instead replace it with uracil. So uracil is equally able to bond with adenine as thymine is. Now, the primary difference, if you were to ask a little kid what the difference between DNA and RNA, most that I know would say, well, one starts with a D and one starts with an R. And the reason why is that they have a different sugar. The deoxyribose is why DNA starts with D. RNA starts with R because instead of having deoxyribose, the sugar is ribose. There's a difference of having a hydroxyl group on ribose and just an H, or in other words, one less oxygen, or deoxy. Again, you don't need to know that, just that there's a slightly different type of sugar. And going back to my analogy, the DNA stays protected, safe in the nucleus. There's only one copy. The RNA is like a disposable copy that can be moved around, can get the cookie batter spilled on it. It's not that big of a deal because it's just a copy. Finally, DNA is long. DNA is like all of the recipe books that your family has. It's all of your genes. RNA typically is only one short part. It's usually the recipe for just one gene. Now, there are different kinds of RNA. It's not terribly important that you know their names, but as we talk about the processes, I will be using them, so it is important that you have just a vague idea of what they are. So the first kind is called mRNA. mRNA stands for messenger RNA, and it's like the disposable copy of the gene. rRNA is the part that's actually going to construct the new gene, and so that's what makes up the part of the cell that does that, called a ribosome. tRNA is the transfer RNA. It's the thing that's going to bring the right amino acid to the ribosome to assemble the proteins. Remember, proteins are made up of amino acids. And then RNAi, we won't really get into very much today, but RNAi is kind of like a proofreader. So if we go back to my recipe analogy, messenger RNA is the copy of the recipe. rRNA is like the kitchen where the recipe is made. tRNA is like the chef that does the making of the protein, and rRNA might be like a taste tester at the end to make sure that everything went well. There's really two main steps. There's transcription and translation. Transcription is making a portable copy of a gene. So imagine your mom or aunt sitting in the kitchen taking a photograph of a recipe. That's transcription. Then imagine your mom or your aunt giving you some instructions. My mom can't help herself. Whenever she makes a recipe, she's always got to give me tips for how to make it. And so these are kind of like tips and processing tweaks that you can make to the recipe that affects the outcome of the protein. We won't really focus on that middle interim part that's basically like editing. The other main part is called translation, and you're familiar with that word in foreign language. When you translate something, you translate it from one language to another. In this case, the translation is translating it from the language of nucleotides to the language of proteins, which is amino acids. So in translation, in the ribosome, the tRNA is going to use the code that's contained in the messenger RNA in order to assemble a protein in the right sequence of amino acids. To transcribe means to make a written copy, and so in this case we're not writing in words, but we are making a copy of the DNA or of a specific gene, and we're copying it in RNA. And we're going to do that using complementary base pairing. And remember that the bases that make up RNA are the same bases, with the exception of uracil, that make up DNA, and so they are complementary to each other. And uracil in RNA can bond with adenine, so it kind of just takes the place of thymine. So transcription, again, is copying the DNA from a template in the nucleus. So the first thing you don't really need to worry about is that there's a promoter sequence. It's kind of like in a recipe book where there's a title in the top of a recipe. The main actor in transcription is an enzyme called RNA polymerase, and just like DNA polymerase built a DNA molecule, RNA polymerase is going to make an RNA molecule. And so RNA polymerase is kind of a multi-tool, if you will, in that it's able to break the hydrogen bonds between the two DNA strands to pull it apart. It's able to unwind it simultaneously, so it's not all coiled and twisted up. It has some other parts that help it, transcription factors and some other things that help hold it open. We're not going to worry about those. So step 1, really step 2 because of that promoter sequence thing. But the first main thing that we need to worry about is that RNA polymerase is going to open up the DNA at the site of the gene. Then, just like it did in replication, RNA polymerase is going to use complementary base pairing. If it knows one side, it knows what to compare. A's go with U's because we're talking, it's going to pair and make RNA. So A is going to bond with U, and C is going to bond with G, so that if you know one side, you can make a complementary sequence on the other. At the end, it's going to get to the end of the gene. It's going to be signaled, hey, the gene's done, everything's copied. And once everything releases, the DNA winds back up. The hydrogen bonds kind of snap back together. As soon as RNAs pass, those hydrogen bonds are attracted to one another, and they just seal back up between the two bases, and DNA is completely the same as it was before. Here's an overview. Let's take a look at some manipulatives. As I model transcription, this area is meant to be inside of the nucleus, because that's where my DNA is. And this structure here is meant to be RNA polymerase. Now, RNA polymerase, as I mentioned, is a multi-tool in that it can do a couple of things. And the first thing it can do, notice the structure, is this point right here in the shape is going to break the hydrogen bonds that is holding the guanine and the cytosine together. And then it's going to get to the active site. Notice the strand on the bottom is just kind of staying out of the way, so that's the top strand that's coding. I've got C here, and these are meant to be DNA nucleotides, so this is an RNA nucleotide, and it's going to come and bond with that when it gets to the active site. And again, I can't have it bond with a C, I can't have it bond with a T or an A. It's got to be the right shape. That's why it's important that the nucleotides of RNA are complementary to the nucleotides of DNA. It's going to move over. Now notice I've got another fork in my RNA polymerase. And here it's going to break the hydrogen bonds, holding the C and G together. I've now got to a spot where I've got an A DNA nucleotide. I don't have any T RNA nucleotides, but I do have a U RNA nucleotide. So this U is complementary with A, so I can bond that there. We'll fast forward now. When the DNA pulls out of the RNA polymerase, the hydrogen bonds reform, and the DNA is completely the same as it was before. The mRNA chain pulls out the other channel, and now we've got a single-stranded mRNA molecule that can then move out of the nucleus and move on to a ribosome. Notice in this model, we will not be modeling mRNA editing. So the product of transcription is a molecule known as pre-mRNA. Essentially, it is a copy of the complementary sequence of DNA. Before it gets translated into a protein, there's an intermediate step that we can just call mRNA processing. It involves two major things. The first thing is that sometimes there's some unnecessary information contained within the gene, and they're removed through a process that we call splicing. It's just a way of making variation and controlling the process a little bit more. When splicing happens, the parts that get removed are ironically called introns, and the parts that get kept are called exons. You don't need to worry about that. It's just kind of a side note. The other thing that happens during processing is that the mRNA needs to be protected. If my aunt was going to take an old school piece of paper and send me grandma's recipe and send it through the post office, she would need to do a little bit of protection. She wouldn't just throw a loose piece of paper in the mail. She'd need to put an address on it, a stamp to make sure it gets there, and then put it in an envelope to protect it. The analogy with that is basically protecting the ends, kind of like burning the end of a rope to make sure it doesn't unravel. So the ends get protective caps on them. And then finally, the mRNA, remember, it's a portable copy, that's its function. So finally, the mRNA can leave the nucleus to go find the ribosome where protein gets made. So we can see the editing process here. The introns, or the unnecessary parts, gets removed. And then on the ends of it, we add a cap so that our final mRNA sequence has a protective end, the coding sequence, and then another protective end, which is a bunch of adenine. So our next step is to actually translate the mRNA, in other words, to make the protein by translating from nucleotides to amino acids. So remember that protein structure has 4 layers. It's folded, it's clumped, different subunits are pulled together, but the whole heart of it is the sequence of amino acids, and that sequence determines its shape and how the protein functions. So when we make a protein, it's all about putting the amino acids in the right order. So the process of translation is decoding the mRNA to string together the amino acids in a specific order. This would be like actually making the recipe. So in other words, turning our genetic code into a functioning protein. So just like everything else we do, we can simplify this as much as possible, but no, in reality, it's always a little bit more complicated than we let on. Our first step is when the copy that we've made in translation, the mRNA, finds the kitchen. In other words, that the mRNA attaches to a ribosome. It attaches at something called the start codon. A codon is a sequence of 3 nucleotides. We'll talk more about that in a second. And the start codon is always the same. It's AUG on the mRNA. Before we can understand the next step, we have to understand the chef. In other words, the thing that's actually going to be doing the assembling of our protein. And the chef in our analogy is a tRNA molecule. As its name implies, it's made up of tRNA, and sticking out on the end, on the bottom, there's different ways of drawing it. In reality, it looks kind of like this, in this upside down L shape. It's often drawn a little bit like that to represent what parts are bonded together. You don't need to worry about any of that. But what you should know is that our mRNA and our tRNA that's going to be doing our active making of the protein have 2 important complementary parts. On the mRNA is something called a codon, and it's a sequence of 3 base pairs of nucleotides. On the tRNA is something called an anticodon, and they're the little nucleotides that stick out to the end, and they are what can bond to it. So your cells can't see. They don't have we talk about the nucleus being kind of like the brain, but they don't actually have a brain and a control system like we do, like our bodies do. And so the way that they control what amino acid comes is through this base pairing. Because GAG is always going to pair to CUC, it will always have a specific amino acid. So we talked about step 1 is the mRNA attaches to the ribosome at the start codon. Step 2, the tRNA is going to pair with a complementary mRNA to bring with it a specific amino acid. And once again, the codon is the part of the mRNA that pairs with the anticodon on the tRNA to control this. So another tRNA molecule comes in and bonds with the next codon, the next set of 3 nucleotides on the mRNA. The tRNA that came has its own amino acid on it, and so a bond forms between the new amino acid and the existing amino acids. It happens to be called a peptide bond, and so sometimes this string of amino acids is called a polypeptide chain. That's not important, I just wanted you to be familiar with the term in case you see it. So then the entire ribosome moves over a little bit. And the tRNA that no longer has an amino acid because the amino acid's bonded to the new amino acid and the new tRNA, that tRNA goes out of there. And that now leaves an empty space as the whole ribosome moves over again. This whole process repeats over and over again, making the amino acid chain longer until eventually a stop codon is read, and a stop codon is just a bunch of tRNA that doesn't have an amino acid on top of it. And so that signals everything to let go, and then we've got an amino acid chain that can go be made elsewhere in the cell, can be folded and clumped into the shape it's supposed to have. So in my opinion, this is much easier to understand when you see it happen. So let's look at that now. Something that happens outside of transcription and translation is the tRNA in our model that they look like this. The tRNA finds amino acids in the cytoplasm and makes that bond there. These amino acids are supplied by our diet. And so by the time translation starts, the tRNA has already hooked up with the appropriate amino acid. So now this big purple thing is meant to be a ribosome, and this ribosome is sitting in the cytoplasm. And our mRNA that we made through transcription has left the nucleus and has found its way to a ribosome. And the ribosome attaches bottom subunit first, and it attaches to the sequence AUG. Methionine comes in and attaches because the anticodon on tRNA, UAC, is complementary to AUG. The tRNA that has methionine in it scoots over to what we call the P site. Don't worry about the P site. Then the tRNA molecule cysteine that is complementary to our next sequence, the anticodon, is attracted to the codon. UGU is attracted to ACA, and that always pairs with cysteine, and that's in the A spot. The ribosome causes a change that causes this to break off and to make a bond here instead. And so now we've got two amino acids bound with something called a peptide bond, and this whole thing scoots over. This guy starts to break away. He's now empty and can go off to find another amino acid sitting out in the cytoplasm. And our next tRNA molecule, which has found its amino acid, can come in the A spot. In real life, our genes are much, much longer than this. But as we near the end of our model chain, we get our last few tRNA molecules in here, and we have our amino acid chain, or our polypeptide chain, coming out the spot on top. Our last codon, in this case it's UAG, this is the stop codon. There's no amino acid up here, and that causes everything to release. And our amino acid chain would float off, our empty tRNA molecules would break apart, and even our ribosome would come apart into two subunits. The really cool thing is a lot of times we need multiple of the same protein in our cells pretty quickly, and so this mRNA chain is completely unchanged. It can go find a new ribosome and move through and make more amino acids, amino acid chain. And this amino acid chain, based on where the attractions are, the hydrophobic and hydrophilic and the acids and bases and so forth, will fold up and clump up to make a protein. And that's protein synthesis. So what I've just modeled is how the cell works. It's all through complementary base pairing. But as human beings that have our own computer, our own brain up here, we don't have to go through quite as many steps. We're able to look at a strand of DNA and determine what amino acid should be coded for it. And here's how that works. Remember, each tRNA molecule has only one amino acid that goes on the end, and one amino acid is based on what anticodons at the bottom. So each anticodon, and therefore each codon, codes for only one specific amino acid. So if our DNA is this sequence, then in transcription we will get mRNA that is complementary to it. What mRNA is complementary to this sequence? Remember, we're making RNA, so there's no T's. So hopefully you got it. Now our cells would use the anticodon on the tRNA. As human beings with a brain, we don't have to. We can use something called a codon chart. So instead of it's called a codon chart, so we're going to use mRNA, not the complementary bases. We're going to save ourselves a little time, and we can plug it into this. So if the mRNA, which I copied from the previous slide, has this, we're going to use the first letter here. So our first codon is UUC. So our first letter in this chart is U. So what that means, it's going to be one of these up here in this column, or this row. The next letter, it's UUC, so the second letter in the codon is U, which is in this row. And so the first row and the first column means it's one of these in this box. And since it's UUC, it's going to be phenylalanine. Take a look and see if you can figure out, using the codon chart, what amino acid GAC will be. Did you get it? GAC is aspartic acid, abbreviated ASP. How about the codon CGU? Did you get that? Arginine, or abbreviated ARG. You don't need to know the abbreviations. Good golly, you don't need to know that. And finally, UAA. Did you find it there? UAA is a stop codon. This is the traditional codon chart. A lot of my students prefer to use this circle one. So we can do the same thing. UUC, we're going to start in the middle. So U puts us in this quadrant. And then the second one is the next ring out. So UU puts us in this section here. And then UU, so UUC puts us here in, once again, phenylalanine. GAC, can you find it? GAC. How about the codon CGU? Can you find it? Down here, CGU. And then finally, UAA, UAA puts us once again at the stop codon. Here's an overview of the entire thing. Again, I think seeing it in picture form is a lot easier than text. So if you want, pause it here to label the diagram in your notes. One last thing before we finish up. Sometimes this process is called the central dogma, and that refers to the flow of genetic information in an organism. Or in other words, going from DNA making RNA and then RNA making protein. And just to review, DNA makes RNA through the process of transcription. And then RNA gets translated to protein through the process of translation. There are lots of animations on the internet. If you're confused by this, I highly encourage you to pick one or two off the list that I post and watch. Most of them are just a few minutes. As always, make sure if you have any questions, you come see me.